Hello everyone, this is Dr. Hira Khalid and I'm serving the Department of Chemistry in Foreman Christian College University as an associate professor. Recently, I have completed my US Fulbright fellowship in the University of Minnesota in Department of Medicinal Chemistry and the ITDU, Institute of Therapeutics, Discovery and Development. Firstly, I really wanted to welcome you all for this event and your participation. And I really appreciate and congratulate the organizers of this VCCA 2024, Professor Ramasamy, the chair in computational chemistry, for conducting such successful events, interactive events virtually and enabling the global participation there. So many of the applauses and congratulations to all of his team members. So today I will talk about the one of the topic, which is the design and the synthesis of triazole derivatives as potential anti-cancer agents. My work is mainly focusing on the three major areas. So one of this is the cancer, and my core focus is for the heterocyclic chemistry. So we have synthesized the hundred of different heterocyclic compounds with the polyfunctionality, with the hybrid molecules, and to examine their biological potential. So today I've selected one of the triazole scaffold and to unfold its potential, anti-cancer potential, what we found in our synthesis and in our group while we have synthesized these compounds and we conducted their anti-cancer screening through the computational tools and through some of the bioassays. So the first, before going into the triazole and their potential and or my findings of in our group, I wanted to demonstrate the journey of this drug discovery in this one slide, because many of the times I got the questions from the students, from the group members, and the people who are interested in the drug discovery and the development or in the medicinal chemistry. So they should be familiar that what is this journey about. So the discovery and making the drug is basically the demanding and multi-scale project. So basically it includes three phases. So this yellow one is the drug discovery, where the chemist, medicinal chemist, synthetic chemist, bioinformatics, and the pharmacist and the cheminformatics, all these people are involved in this phase. And this phase is the most expensive, the crucial, and the time-taking phase in the drug discovery and its development. Then there is the clinical testing, which is the blue one, which include the phase 1, phase 2, and phase 3 clinical trials before bringing it into the approval and the market. And then there is finally the approval for production. So once the phase 1 and phase 2 and phase 3 passed and there is a success in these phases, so then this goes through the FDA review and approval, and then there is the post-approval. And finally, once the FDA approved this one, so the drugs comes into the market for the recommendation, for the prescription, and for the users of the consumers or the patients. Now, you can see that this drug discovery include the target identification, which is that which enzyme, which protein you are targeting or which receptor you are selecting for making these compounds, and they will inhibit that activity to work as the anti-cancer or any of the drug in disease in you are interested. So its activity So first thing is the target identification. And here, this identification we do involve a lot of computational resources. And here we do have the opportunity that we can go for the screening in-house compounds if you already have. Then we do the available data bank, and the one of this one is the zinc data bank for the screening of the millions of the compounds. Then from the screening, when you through the docking and the binding interaction studies, you find out that, okay, this type of compound structure would be potential to synthesize in the lab and then to test to the bioassays. So it leads to the lead discovery. So out of million of the compound, then you chalk down, you narrow down the millions to the hundreds and then the tens, and then you selected the 10% of these to further go into the lab and synthesize that one. So once that compound has synthesized, that has characterized, so it involves your reaction optimization as well. It also involves the lead optimization because the solubility, lipophilicity, log p values, the Lipinski 5 rules. So these are all of the consideration which you have to make and you have to adjust for finalizing the lead optimization. Then there is the in vitro, through the in vitro testing. Then you go for the in vivo testing, which is conducted on whatever the models we will use. Either we are going for the mice, rat, or on the dog, or any of the tested animals for these preclinical testing. So this involves the years of investigation. So you might have seen, if you have started or you already working or you are aiming for working in the medicinal chemistry. So it took your PhD years, 5 to 6, and the 10 years for getting into and completing into this drug discovery phase 1 process. So it also involves the amount money it is, because it's costs around the 600 millions to 800 million dollars, and because you need the resources for the synthesis, you need the resources for the screening, you need the test for the toxicity testing, for the bioassays. So it all requires the budget for conducting these working. So the next is, of course, a little bit less years, and of course the phase 2 is also the one of the expensive, and this is the most expensive in that term, that it costs the billions, because then you are involving the living species and the humans into the phase 1, into phase 2, and phase 3. So here, this is the idea that from the drug discovery, we get the 200 entries that we selected the preclinical testing 200 compounds. And then after doing this one, we just get out of 200 that, okay, these 5 compounds are fulfilling that criteria, and we can go for the phase 1, phase 2, and phase 3. And then after doing this one, we just approved the one by the FDA. So you can see that how demanding and how much extensive is this drug discovery and the drug development approaches. But at the same time, it is very rewarding and satisfactory that you're contributing to the human health for the well-being, and you're learning in the process a lot of different skills, and do, undo, and then revisiting your work. So this is the important about the drug discovery cycle, that how any of the drug, when it is in a market, so what are the different steps it has undergone. Now, the next thing is coming to the main topic. So the 1,2,4 triazoles, though they are also available in another isomeric form, which is a 1,3,4 triazoles. So why we selected the 1,2,4 triazole to work on? So there is a very strong literature foundation that it has the broader range of biological activities, which include the antifungal, antibacterial, analgesic, anti-cancer, antidepressant, anti-HIV, antiviral, anti-TB, anti-inflammatory, and antioxidant activities. So with this literature foundation, and there are the very good scientific researchers about the 1,2,4 triazole as the anti-cancer agent. So working on the different other heterocyclic nitrogen-based heterocyclic compounds. So we picked the 1,2,4 triazole to work on and to examine its anti-cancer potential with the different functionalities which we are introducing with merging with that scaffold. On the parallel line, we were also working on the 1,2,4 triazole as the anti-Alzheimer drug agent, and we have the very good and the motivating and the promising results from that one. So the work I'm presenting in front of you is not yet published. So that's why some of the results I may not show here because we are just submitting for the patent and the publication. But the maximum I'm revealing is that how we have synthesized and what are the different potential what is the anti-cancer profile we get through the computational tools. So by having this foundation, we worked on this 1,2,4 triazole. In the next slide, I will tell you that what was our scheme we designed for synthesis of these derivatives. But firstly, I wanted to give you the idea that the triazoles are basically the 5-membered 3 nitrogen-containing scaffolds. And there are many marketed drugs which contain the 1,2,4 triazole group. So I picked 3 of them to give you the idea that this triazole ring in a compound structure, and how just with the minor modifications, you can see that there is this 7-membered ring as well, and they are very similar to this one. But we know as an organic chemist that the slightest modification can change the biological activity and the behavior of that compound. So I have selected the triazolum, aprazolam, and atazolum for your reference. So this triazolum is useful for treating the insomnia, while the aprazolam is for the anxiety and panic disorders, while this atazolum is a drug which is used for the muscle as a muscle relaxant and anticonvulsant. Why we are focusing on the cancer? So the prevalence of cancer is hitting globally, and still we are fighting. Even there is abundant of research is going on, but there are the mutations, there are the epigenetics which are involved, and there are the resistance. There are still a lot of things to do to work on for fighting the cancer. So the cancer mortality, so there are the different types of the cancer. So some of these I have listed, and with the perspective of what are the most common one in the women are the breast cancer, lungs cancer, colorectal cancer, and there are the other types of the cancers, ovarian cancer as well. So these are the different types of cancer which had the major mortality, which is causing the major mortality in the women. So through the researchers, the receptors which we are working for the 1,2,4 triazole are the DNMT, and they are having a role in the breast cancer, the lungs cancer, colorectal cancer, and even the ovarian cancer as well. So that is our objective, to work on this and to explore that how we can contribute through the synthesis of these new drug agents, and we can mitigate for to the cancer, and to see the potential of these compounds with the different cell lines. Now the overview of DNMT1 and DNMT2 and their significances in the cancer treatment. Now the first thing is, what's the DNMT1, what's the DNMT2? So these are basically the two enzymes which belong from the enzyme family, which is the transferases, because it is responsible for shifting the one functionality to another one. So if you go to the different enzyme classes, so this is one of the major enzyme class, which is the transferases. So the name states DNMT1 is stands for the DNA methyltransferase 1, and there is another one, DNA methyltransferase 2. So we selected these two receptors for our enzymes to see that either our enzymes do inhibit these one, and if they do, so to what extent. Why? Because they are the major contributors in the cancer projections. So DNMT1 is the enzyme which is primarily responsible for maintaining DNA methylation pattern during DNA replication. It adds methyl group to the 5th carbon position of cytosine residues in CpG dinucleotides, while the DNMT2, it has a weak DNA methyltransferase activity, and primarily it methylates cytosine in the transfer RNA. Its role in cancer is less understood, but is considered important for the RNA methylation, while the DNMT1 role is much studied and established one. In this slide, you can see the comparison of the DNMT1 and the DNMT2, their 3D structure. And here you can see that there is this basically concentrated into the nucleus, and this is transferring the methyl group to the DNA, while the DNMT2 is giving the methyl group to the transfer RNA. So this is the basic difference between the DNMT1 and the DNMT2. And in the next slide, you would be able to see its occurrence. So the DNMT2 is basically existed in the nucleus as well as in the cytoplasm. And here you can see that the transfer of the methyl group is happening to the transfer RNA, while the DNMT1 is present in the nucleus, and it basically contributes for the methyl transfer to the DNA. And then the DNA, through its replication and the translation to the different steps, and this DNMT2 would be responsible for this methyl group transfer to the transfer RNA. So the both are basically have the role in the projection of or the propagation of the cancer cells. So the role in cancer, the epigenetic regulation, both DNMT1 and DNMT2 are involved in the regulation of gene expression through epigenetic modification. So the epigenetic modification involves the gene mutations. It affects through the environmental factors. So when this is happening, so through the regulation process, so this is adding up into the new generation. So the DNA methylation, primarily mediated by DNMT1, is a crucial mechanism for gene silencing. Tumor suppressor gene, aberrant methylation of tumor suppressor gene by DNMT1 can lead to their silencing, contributing to tumorigenesis. This hypermethylation is a common feature in many cancers. Oncogenesis. DNMT1 can also contribute to the hypermethylation and activation of oncogenes, further driving cancer progression. So here you can see that how much this DNMT are contributing into the cancer, and we can use it as the biological markers. So, or you can say the chemical markers for the prognosis and for the diagnosis of the cancer. So, as I said, the DNMT inhibitors, the drugs that inhibited DNMT1, such as azacitidine and decitabine, are used in the treatment of certain cancers like myelodysplastic syndromes, MDS, and acute myeloid leukemia, AML. These inhibitors demethylate DNA, reactivating the silenced tumor suppressor genes, which were silenced and they were not able to suppress these tumors from the projection, from the growth. So now it reactivate by demethylation the DNA. So that means that we are inhibiting the DNMT activities, that we are stopping there for transferring this methyl to the DNA. For the cancer therapy, the other approach which is used is, along with the DNMT inhibitors solely, the other approach is the combination therapies. So the DNMT inhibitors are often used in combination with other therapies to enhance their effectiveness. For example, combining the DNMT inhibitors with histones, deacetylase inhibitors, HDAC inhibitors can have synergistic effects in reactivating silenced genes. So the DNMT can be used as the biomarkers for cancer detections and the prognosis. This is a potential field to explore and work. The DNMT2 role in the cancer is less known, and there is a need to understand the functions which could reveal the new therapeutic targets or the biomarkers. Secondly, there is a need to develop the new, more promising DNMT inhibitors. So these next generation inhibitors aim to improve patient outcomes and expand the use of DNMT inhibitors to a broader range of cancers. Now coming to the which scheme we followed for the synthesis of these triazole. So here is a very interesting organic reactions are involved. So we started with the simple molecule and we used the organic acid. Firstly, we convert it into the ester, so through the Fischer esterification. Then we convert it by the reflux reaction and with the hydrogen hydrated. So that was converted into the hydrazide. And then this was reacted with the isothiocyanate. And here we have used a different isothiocyanate, but here for this particular series, we picked the 1-chloro-4 isothiocyanate to benzene because these halogen substitutions are also playing the crucial part in inhibiting and the DNMT, and they are showing the promising biological potential. Then, firstly, we get the uncyclized triazole, which was then reflux in with the 10% potassium hydroxide, and we get our parent compound, which is the triazole thiol. So here from this acid side, we have played around this molecule to introduce the different substitutions and the other functionalities The amide functionality is also one of the um, good functionality which enhance the binding with the other side of the amino acid residues, because it has a carbonyl, oxygens and the amino group. So they are the hydrogen bonding, these electrostatic interactions which help to get this strong ligand and enzyme complex. So here we move on to the two directions. So we also see the different alkyl chain substitutions. We also did the acetamide substitutions, and all of these compounds we generated in our lab started from the very simplest organic acid. So many of our 1,2,4 triazole compounds has been published, their synthesis and their biological potentials. One of the representing publication is in Turkish Journal of Chemistry, and you can also see the Pakistan Journal of Pharmaceutical Sciences, and there are other more. Now, these are some of the structures from this series of those compounds. So HABR1, HABR2, HABR3, HABR5, and HABR4. And here you can see that these all are basically the acetamide derivatives of these 1,2,4 oxazole. So they are, some has the one halogen, one has no other substitution on the phenyl ring, some has a nitro, and some has this in nitrogen, which is also having the ethyl group substituted there. So HBr5 was one of the promising, all of these, but the HBr series was, all of these were the significant one. I will show you in the next table. Another series of this one, which started from the phenylacetic acid, and here you can see this is having the alkyl substitutions and the acetamide substitution as well. So these were the four which were promising from that series of that compounds. And in the next slide, I will show you their computational results for these two receptors, the DNMT1 and the DNMT2, which I just mentioned. And here you can see, so we tested these. I have mentioned the structure, the tier 1, tier 2, tier 3, tier 4, because these are the derivatives. But we also tested these for their starting precursor compounds as well, to evaluate and compare their potential. So out of this, you can see that the tier 4 was the outstanding from these four. And here in the table, what we have mentioned here, so this EB is giving you the idea about the binding site, binding interaction. So more the negative is the good score, binding score, while the Ki in the nanomolar is representing the inhibition constant. So this inhibition constant is basically giving the concentration of the free ligand from the ligand and the receptor complex. So it means the lesser the free ligand, it means you have the more stronger ligand receptor complex. So here you can see in a T4, it is just in a decimal and it's a 0.1, and here this is a 2-digit. But when you see that the binding energy has dropped, and here these are into the 5 digits, this is too high. It means that it is not having the same, it has not have that strong enzyme and the ligand, or the receptor and the ligand complex, which is necessary for this enzyme to work on that ligand and converting into the product one. So the next part is for the series HABR and this one, and from this one we see all of these four were the significant one. And from this, if you just look at this one, so the HABR5 is the having -13, right? And here you can see that it also has the very single to double digits in the Ki, which is the inhibitory inhibition constant as well. So these was very promising results, which help us and which motivate us to proceed further for this. And here, these are the more detailed picture for giving you the idea about the what type of interactions we do have with the different residues of the amino acids of this protein. So these dark green are representing the hydrogen bonding, while the light green are showing the van der Waals interactions. There are the pi-pi stacking, there is the electrostatic interactions, this is the pi anion. So there are a number of different interactions which help the to form this ligand and receptor binding and making it significant, and then to proceed it for the inhibitory activities. So this is the interaction of the TA4 with the DNMT2, and the first one was the interaction of TA4 with the DNMT1. In the next slide, I am mentioning the most promising one, just with the constraint of the time. So this is the binding interaction of HABR5 with the DNMT1, and here you can see the significant hydrogen bonding, the pi-pi interactions, and the van der Waals interactions as well. Next is the HABR of 5 with the DNMT2, and in the next slide I will give you this slide to show that we conducted, because once we established that it is showing the anti-cancer activity, but this is also important that we have to conduct the toxicity analysis, MTTSA, that either our compounds are toxic to the normal cells or they are specific for this one. So this was a very encouraging result that only the HABR1 was the compound which showed the toxicity to the normal cells, so human cells, but the rest were the safe to the toxicity profile. So on the y-axis, this is basically the toxicity concentration, and here these are the different compounds which we have used. So the blue one is showing that we have used these compounds into the two concentrations, 50 micromolar and into the 10 micromolar. So the thing was that the HBr1 was toxic even at the lower concentration, the 10 micromolar. So we are not proceeding this anymore, but the rest of these are the good, safe, and they are under the anti-cancer assays on the different cell lines. We are testing these for the ovarian, breast cancers, and the other cell lines as well. So at the conclusion, DNMT1 and DNMT2 play a significant role in the epigenetic regulation of genes expression, with DNMT1 being particularly crucial in the context of cancer. The inhibition of DNMT1 has proven to be a valuable strategy in cancer therapy, leading to the reactivation of silenced tumor suppressor gene and offering a promising approach for treating various cancers. Ongoing research into the functions and therapeutic targeting of these enzymes continues to enhance our understanding and capabilities in cancer treatment. Now, this is my research group, and they have passed, and these were the people who majorly work in that batch. But this triazole series which I presented, so the LIBOR and the Hafsa was the MPhil student, and the LIBOR was the BS student who worked on that project. And then I want to acknowledge the Department of Chemistry, the Fulbright, to give me the opportunity for extending from the more general approach to the more specific, because now we are working on specifically on the ovarian cancer project. Department of Pharmacy, FCCU, for the collaboration and for conducting bioassays. ORIC for the initial funding and the grant for seeding these projects, and the University of Pescara, Italy, who are conducting the anti-cancer activities and the MTT assays for this project. Now at the end, this is the building, science building of our FCCU. This is where our chemistry science block is there. and it's present in Lahore, situated in Lahore, Pakistan. So you are all welcome to visit and explore. And the FCCU is the first liberal art university in Pakistan. Now, at the end, for all of the listeners and the attendees, I wanted to present the quote of Ben El-Faringa, the Nobel laureate on the molecular machines. So the beauty of chemistry is that I can design my own molecular world. So there's always proud to be a chemist. Is always proud to work in the sciences, whatever the field you